Series, Fish and Fisheries. And even though it's not, we know we're always up against when the weather's good. It's always the case. But I think you're in for special treats. We've been waiting to have Dr. Lawson, Gareth Lawson, for a couple years. We finally could get his schedule to converge with our seminar series. Gareth completed his PhD at MIT Joint Woods Hole doctorate program, and his master's work was at Memorial University of Newfoundland on cod, and he told me cod using acoustic methods. And he basically accomplished his bachelor's degree at McGill University. So he is a Canadian, like our postdoc Angela, who's here. And also I want to point out that Gareth's work, not just on krill, but other, let's say, small things that are of great importance to not just small pelagic fishes, but large pelagic fishes. As you probably heard, giant group and tunic
I don't
krill.
have her mic, so
krill
can you just stand a little closer to her?
Oh, I see. That's what
something that's important to ecosystems and fish. But I also really, really advise you to check out Gareth's blog and his research group's blog. He told me his wife helped him name it with Charismatic Microfauna, and it includes some really cool video of pteropods. And so we're really psyched to have him here. And the title of this presentation tonight is Plankton in the Gulf of Maine, just how important are krill to New England fisheries. So welcome, Dr. Lawson.
Thank you very much. Thank you very much for the introduction and for inviting me here. Thank you all for coming. So like Molly said, I'm a scientist at the Woods Hole Oceanographic Institution, or WHOI as we call it. That's a bit dark, but I think you can make things out. So we're a non-profit, private research institution. We're very much focused on the oceans. We don't have an English department or a history department. And I thought I'd start with a quote from Alfred Redfield, who is an eminent oceanographer at the Oceanographic at the beginning of last century. The photograph here shows Woods Hole, and my building is here. That's the ferry terminal. Those are some of our boats. And my building is actually named after Redfield, and this plaque is on the front, so I see it every day as I walk in. And you can't see the quote there, but I've reproduced it there, which is, "Life in the sea cannot be understood without understanding the sea itself." And that's the idea that pervades WHOI, that we have to think about things as the ocean is a complex, three-dimensional, very dynamic environment, and to understand the animals, the populations, the ecosystems, we need to be mindful of that. So oceanography is, of course, a very interdisciplinary kind of a science. So a little introduction to my overall research interest. You can't see this so well, but I study plankton. So the word plankton comes from planktos, which is a Greek word for wanderer. So the plankton are the things that drift around in the ocean, subject to the vagaries of the currents. And a sort of a long-standing problem in oceanography concerns how these little plankton are distributed in the ocean. The original hypothesis at kind of the end of the 19th century was that they were just evenly distributed, uniformly distributed everywhere in the ocean, the same amount of plankton. And obviously that proved very quickly to be false. There are places where plankton are very abundant, places where they're very sparse. So one of the sort of motivating questions in my work is trying to understand what determines the distribution of these little things. And to illustrate what I mean, this is a photograph. It's a bit hard to see, but it's an aerial photograph taken in the Gulf of Maine during a whale survey. And these sort of wispy features that you see here are actually surface aggregations of krill, so a crustacean that I'm going to be talking about a lot in this talk. And what should jump out at you is that these are far from uniform structures. And so the question is, what leads to these aggregations? Why are the krill here and not over here? And the answer is going to be a sort of complex interaction of the currents, the physical oceanography, and then the biology, the behavior of the animals, their reproduction, how much food is availability to them, and so on. So it's a complex problem. It's a somewhat esoteric one, and part of what guides our research at WHOI is just sort of pure pure science, understanding how the oceans work. But it's also a problem that's important because these animals are prey for various kinds of higher predators, commercial fishes, marine mammals, and so on, animals that we value. So if we want to know something about the distribution of higher predators, it helps to know something about the distribution of these little things that are their prey. Then one of the other things we think about a lot these days is the impact of climate change on the plankton, and that's another topic we work on in my lab. And then the final topic has to do with the development of sampling technologies. So the very traditional way to study plankton is with nets, plankton nets. And you can imagine if you used a plankton net to sample this feature here, that you'd be sort of scooping up a whole lot of these krill, and you'd gain an understanding of kind of the average level of abundance of these little bugs. But you wouldn't be able to learn anything about these very fine features that you see here. So to address those kind of very small scale features, we turn to other kinds of technologies. So shoot.
Can you tell me what the scale of that is?
Yeah, well, so these are seagulls. So the scale of these features are going to be on the order of hundreds of meters, sort of thing. Yeah, they can be bigger. And jump in with questions at any point. This can be informal. And if I'm talking too quick or too, shoot. Yes, okay, good. Thank you for saying that. So the definition of plankton is that they drift, so that they can't swim well enough to overcome the prevailing currents. In the case of these krill, one of the things that's interesting about them is that they're sort of between being plankton and fish. You know, they can swim fairly well. They're sort of 3 centimeters. The largest species in our local waters is about 3 centimeters. So they can swim well enough that they can overcome some currents, and they do sort of aggregate for social reasons, for spawning and to avoid predators and so on. So they're kind of neat because the currents influence them much more than big things like fish. But they're still not quite plankton. Okay. So, and I forgot to show that. Well, you can't see that. So this is just a photograph of a whale chowing down on some of these krill aggregations, reinforcing this idea that they're important prey. Okay, so I was talking about technologies. Much of the work that we do in my group involves the use of acoustic technologies. So basically very fancy fish finders. And the principle here is that we're sending a pulse of sound out into the water column, which is reflecting or scattering off of the various animals, plankton, fish, in the bottom. And then we make measurements of the intensity of the echoes that we get back, and we use those measurements to tell us something about the numbers and the kinds and the sizes of animals that are present. So the nice thing about sound is that it propagates very efficiently in water. So light attenuates quite quickly in seawater. If you're a diver, you know that you can't see very far. Sound you can send out to very large ranges, so you can sample to long distances. And we can send these pulses out at very high frequency, very fast as our ship is moving through the ocean. And the kinds of data we get look a bit like this. This is an echogram, so we're looking at depth, and then this is distance as the ship is going off of George's Bank, so that's the bottom there. And then this is basically a slice through the ocean where the color scale is showing the intensity of these echoes we get back. And you can sort of think of it as being loosely an indicator of how much biomass of animals there are. So where the colors are sort of blue is low levels of echoes, and where it's red is high levels. And so what we can see are these things like these layers, which we think are krill, and then these aggregations here at the edge of George's, which are herring. So we can see both the predator and the prey at the same time. We're sampling to 200 meters, so very long distances, without disturbing the things that we're trying to study. If we put a net in there, we disrupt these aggregations.
So these are present at all depths versus like the top 20 feet or something?
So the measurements or the krill? The krill, they migrate up and down, actually. So they will come up all the way to the surface. Generally speaking, they go down deep during the daytime to avoid predators that use vision. So they go down where it's dark. And then they migrate up to the surface to feed at night. They do often have sort of preferred depths associated with particular temperatures that they like as well. Okay, so we work on a bunch of different sort of planktonic and small animals in my lab, and what we look for is animals that are interesting in some way, ecologically or behaviorally. We usually focus on animals that are important as prey for things like fish and marine mammals. And then we do also look for animals that in some way lend themselves to being studied with these fish finders. Not all animals do. So these are photographs of the kinds of animals that we tend to work on. And a game that I like to play when I give this kind of talk is to point at pictures and have people guess at what they are. All right, so we got the list of species groups here, and I'm going to point, and you're going to guess. Okay, so we'll start easy. All right. So anyone know what kind of species of fish? Yes. All right, so let's move. You can't really see what this one is, so I'll give this one away. This is a siphonophore, which is a gelatinous animal, and it actually has a little bubble that it uses for flotation, and that bubble is a very, very good reflector of sound, so they light up our sounders. How about this one? Yep. Copepod, which is a small crustacean. Well, some of them get to be fairly big, I suppose a couple millimeters. And then how about that one? Yes. So pteropod. Ptera means wing, pod means foot. So these are snails, basically, also known as sea butterflies, because the foot structure that in a terrestrial snail you're probably familiar with, it's evolved in these guys into this bilobed wing, and they use this to flit through the water, so they look a bit like a butterfly, hence sea butterfly. So yeah, these are pteropods, and then this one, process of elimination. Very good. Much better than our communications group last time I gave this talk. Okay, so the main themes of our work currently in my lab are we're studying krill in the Gulf of Maine, which will be the focus of this talk. We are also doing some work on ocean acidification, which I don't know if you're familiar with this, but it's thought of as being the sort of evil stepsister of global warming and additional consequence of anthropogenic carbon dioxide emissions, and the effects of ocean acidification on these pteropods. Depending on how the timing works out, I may come back to that later. And then we're also doing some work where sort of traditional oceanography is done from ships, of course, but we're starting to supplement that with observations from other kinds of platforms. Robots, basically, so vehicles like this torpedo-shaped vehicle that can operate independent of people and of ships. So we're doing work trying to get these sonar systems that we use for observing plankton onto things like these vehicles, and more recently we've actually started deploying them on animals like that elephant seal that you see there, which is a little bit more of a kooky project, but it's fun. Okay, so just a quick outline of where I want to go in the talk. Start with some background, trying to review what's known about krill in our local waters of the Gulf of Maine, and make the case that we don't really know enough about them, and obviously we need to study them more, and then move on to some insights that we've gained from looking first at existing data sets and then some field work that we've done recent. I'll end with some conclusions and comments about where we're taking the work. And of course, this is the output of a collaboration with a number of scientists at WHOI, as well as students and technicians, so I need to acknowledge them here. Okay, a little bit on krill. Technically, the formal name for this group of organisms are euphausiids. Krill comes from the Norwegian word krill, but with one L, which apparently means fish fry, and I'm not quite sure how that got conflated with these little crustaceans, but I think it has to do with both of them being prey for whales. So if you're familiar with krill, it may well be from the Antarctic, where they've been very well studied, and they're known to be sort of the cornerstone of the ecosystem. So what we have here is the Antarctic food web, a little schematic. So we got our krill here. They're feeding mostly on phytoplankton, which are the plants of the ocean algae. They also feed on small plankton like our copepods that we saw a minute ago that are herbivorous. And then the krill themselves are prey for all the cute things that we associate with the Antarctic, the penguins, the seals, and whales, and so on. They're also prey for humans. So there's a very large krill fishery in the Southern Ocean. It's the largest crustacean fishery in the world. Some of it is for human consumption, like you see here. The majority of it is actually for fish meal. for feeding to aquaculture fish. So the salmon that you might see from Chile, most of that has been fed Antarctic krill, partly because it gives the flesh that nice orange color that the consumer really needs. So in our local waters, though, in contrast to the Antarctic, we know a whole lot less about krill, but we have reasons to believe that they may be a very important ecosystem member. So what I'll do now is just tell you a little bit about what we do know from studies of the Gulf of Maine, starting with Henry Bigelow. So Henry Bigelow was the first director of the Woods Hole Oceanographic Institution. He was the founding director. He was instrumental in causing the oceanography to come about. He also was a strong advocate for our first research vessel, the research vessel Atlantis, and that's Bigelow at the helm of the Atlantis. The Atlantis is the ship that you see in our logo, and is also the namesake for the space shuttle at Atlantis. So Bigelow, at the early part of the last century, did one of what remains one of the most comprehensive series of surveys of the Gulf of Maine, looking at the hydrography, the currents, the properties of the seawater, as well as the animals. And what he found for the krill remains one of the best sort of descriptions we have. He published this in 1924 in a monograph called Plankton of the Offshore Waters of the Gulf of Maine. And what he found was these krill were widely distributed. Are we all familiar with the geography of the area? I can review. So we have Cape Cod, Woods Hole's down here. We're right here, Cape Ann, Nova Scotia. And then this is George's Bank. And that's the edge of the continental shelf kind of there. So these dots are where he caught krill. And so he caught krill kind of all over the Gulf of Maine and saw some suggestion of a seasonal shift. So in the sort of July through early fall period, they were mostly in coastal areas or at the edge of the continental shelf. Whereas by sort of February through April, it had contracted and they were mostly in the sort of deep basins in the central Gulf of Maine. So aside from this kind of just description of where Bigelow caught krill, we don't have that much information on these animals, but we do have these sort of tantalizing glimpses that suggest that they really are very important. And one of those comes from the stomachs of whales. So back in the days of whaling, there's a group of people who studied the stomach contents of fin whales. And these fin whale guts were just chock full of krill, which suggested two things. One, that the krill were very important, but the other, that based on what they thought were the sort of energetic needs of the fin whale over the course of a day, and how fast a fin whale can swim, that the krill had to be very patchy in their distribution. They couldn't be just sort of distributed at these sort of even levels, background levels that were sort of thought to exist at the time. They had to be clumpy in their distribution for the whales to be able to make a living off of them. They wouldn't be able to swim fast enough if they were broadly distributed. So the inference is that krill can be important, at least for fin whales, and that they must be patchy in their distribution, and that patchiness influences their availability to the predator. And then since then we've had sort of these few observations, and I should say these are observations in the published scientific literature. They're observations of krill by other people all over the place. But these squares, these red squares, show places where people have published on their observations of large aggregations of krill, usually made in conjunction with sightings of various kinds of top predators, seabirds, commercial fishes, often herring, and often marine mammals. So places like the Bay of Fundy, where there are what seem to be spawning aggregations of krill, and then at the margins of some of the deep basins in the Gulf of Maine, and then also along the southern flank of George's Bank, especially in the canyons. So the question that sort of motivates me is well, what leads to these aggregations? What drives that distribution? Then the other sort of motivating factor is as you probably know, there's a certain movement in fisheries management towards what's called ecosystem-based management. So rather than managing individual stocks, individual fish species, sort of recognizing the interconnectedness of the ecosystem and managing it as a whole. And if we're going to do that, we have to know more about these little things that are at the base of the food web. we also need, of course, very good models, simulations of how the ecosystem work. So part of what we're trying to do is help collect some of the data that will inform that modeling. So there's sort of an ecosystem management component to the work, and then there's also the fact that not only are there fisheries for krill in the Southern Ocean, there's actually been a test fishery in the Gulf of St. Lawrence. It was some years ago, but I heard from colleagues just recently that it's restarting. There's going to be a commercial fishery for these tiny little crustaceans. And a lot of that is driven by the dietary supplement industry. So these can be sold for a lot of money, krill oil. So we don't have a krill fishery in the Gulf of Maine as yet, but there may be one day, and it sort of gives us an opportunity to study it pre-exploitation, which is kind of a nice position to be in. Okay, so that's kind of what motivates the work, and the objectives in this sort of krill part of the research program are to understand what is driving this krill distribution at large geographic scales over long time periods, and then right down to sort of small, short time periods and small scales. And the goal is to really understand what the ecological role of the krill is, how it interacts with these commercial fishes and other species. And then the other part of the research program involves developing these accurate methods for sampling krill, because our argument is that the reason that krill have been understudied and underappreciated is that they're very difficult to catch. So your question about whether they're agile or whether they're planktonic, they're agile enough that they can get out of the way of these very slow-moving, small mouth opening nets that we tend to use. So they've been, our argument is that they've been drastically undersampled. So we've been working on resolving that sampling issue. Can't see all these photographs, but the approach we've been taking is to use a variety of sensors, not just nets, but other things. So we're using the acoustics that I mentioned before. We have fancy net systems that I'll come back to. This is a video camera system. You can't quite see. It collects images of the krill. And then we relate what we learn about the krill to data on fish that we get from our collaborators at Northeast Fisheries Science Center. And then when we do our own surveys, we bring along a team of observers who count the marine mammals, seabirds, and other surface-associated things. Okay, so I'm going to move on to talking about some insights we have from looking at existing data sets. And the two main data sets we have are first zooplankton samples that were taken. Zooplankton, pardon me, I should say, is the animal component of the plankton as opposed to the phytoplankton. So these are samples that were collected by Northeast Fisheries since 1977 using bongo nets, which is this net system here. It's actually got two nets that are connected, so it looks a bit like a bongo drum. It's a terrible way of catching krill, but it still gives us sort of a relative indication of how many krill are there and helps us understand their geographic distribution. And the other thing we look to is what's called the food habits database maintained at Northeast Fisheries. So basically, as you probably know, there's a bottom trawl survey that happens a couple of times per year for stock assessment purposes. And since '73, they've been taking subsamples of the fish and looking to see what's in their guts and quantifying and enumerating that. So they find a lot of krill and they record that. So we can use those data for a few things. We can use it to look at the geographic distribution of the animals. So here we have a map. There's Cape Cod again, Georges Bank here. And what you're looking at is each one of these dots is a place where there was a net tow that caught krill. And the size of the dot means more, bigger dot means more krill were caught. So in the springtime, you can see a certain concentration here at the shelf edge and also in coastal areas. And then we can also look in the stomachs. So the stomachs are great because we can see where the interactions are happening between the fish and between the krill. So again, the dots indicate places where a fish was caught with krill in its guts, and the size of the dot, and the more red the dot, the more krill. So you can see there's not much predation on krill happening on Georges Bank itself, but at this southern flank at the edge of the continental shelf, there's some activity, and then here on the sort of northwestern side, there's a lot of interactions happening, and that's mostly herring chowing down on krill. So we can also look at the importance of the krill in the diet of the various fish that tend to feed on them. So this is one of the few graphs I'm gonna show, but what we're looking at is for a variety of fish: dogfish, skate, herring, silver hake, pollock, red hake, and redfish. We're looking at the variability in the proportion of diet made up of krill. So to help you interpret the graph, for redfish, for instance, what this means is that in some years, as little as none of the diet is made up of krill, 0%, and in other years as much as 100% of the diet, with an average of about 30%. So the take-home message is that there's a lot of variability between years, between species, but in some years, some fish species, krill, are more than 3/4 of the diet. So certainly they seem to be very important, and we want to poke at this problem in more detail. Okay, so let's move on to some recent field work that we've been doing. We've had surveys, cruises targeting with the locations sort of guided by looking at those existing data. We've had cruises targeting this region here on the northern flank of Georges. And what I'll talk about mostly here is work we've been doing here south of Cape Cod at the continental shelf break. So just a...
What's the size
the krill? Well, there are actually a variety of species, so some of them can get really quite small, but the largest species, which I think is probably the most important one for the commercial fishes in the Gulf of Maine, is about 3 centimeters, inch and a bit, as an adult. They're small.
Because their mobility is low, they pretty much stay in the Gulf of Maine year round, or does spring and summer variations, like when the Gulf Stream comes up, to triple the size?
Excellent question. Excellent question. And we don't have enough data to really know. So I can't answer, unfortunately, at this point. But I would love to know if there's almost certainly import of krill happening from the north and coming in through the Northeast Channel. We see a lot of krill down here as well as here. So maybe it's suggesting that there's sort of a conveyor belt of krill through the region. We also don't know much about the sort of reproduction. They spawn here, but do all of those offspring then get washed out, or is there sort of retention? Open question. Pardon me?
When was it taken, this data?
Oh, these. So these squares are reports that started in probably 1978, and the most recent would have probably been about 2004. So there have been these sort of just few papers every now and then of people saying, "Oh, we saw a lot of krill right there." Yeah, yeah. Well, yeah, exactly. And you go back year to year in some of these spots, and the krill are persistent. Pardon me. I should correct myself on that. At these canyons here, when they first observed them, and they were in a submersible, and they saw these huge schools of krill, and they published it in Science magazine, so very high-profile journal. All these krill, and they said, well, we think that maybe this is subsidizing Georges Bank, that the fish, because many of the fisheries are, of course, prosecuted on the continental shelf, so maybe some of those fish are going to the edge of the shelf, going into these canyons, feeding on the krill, and then taking that energy back up onto Georges Bank, which was a very provocative idea. They went back the next year, same submersible, no krill. And that might be part of what is driving all that variability. There seems to be a fluctuation on so I haven't done a statistical analysis, but by eye, I would guess sort of 5 to 7 years sort of cycle of when the krill are abundant, when they're not abundant. What drives that? I don't know. Climate, perhaps? Yeah. Temperature, yeah.
How long do they usually live?
These ones are on the order of a couple of years, but the aging is quite difficult. In the case of the Antarctic krill, they were thought to be sort of 2 to 3 years, and it turns out they can live to 6 to 7 years, but they shrink. So, yeah. Okay, so well, you can't see any of the bathymetry here, but so what you're kind of looking at here is the continental shelf edge, and the current structure, the physical oceanography of this area is extremely dynamic, and the physical oceanographers at WHOI are very excited about this area, and to a large extent, the biologists sort of follow where the physicists lead. It's interesting, or it's because it's sort of a frontier between water masses, between the shallow and generally colder waters of the continental shelf, and then the warmer, saltier, deeper waters of the continental slope, and then ultimately the open ocean. So you have a shelf current that flows along the shelf towards the southwest, and then the Gulf Stream, of course, flowing to the northeast. And then there are these interesting eddy features, these warm core rings. So you have meanders in the Gulf Stream. You can pinch off, form these eddies that persist for a year or more, and move their way along the shelf break. So from the standpoint of the currents, the edge of the continental shelf is a very it's a hot topic. And from a biological standpoint, we don't know very much about the area. We know that it's very high productivity. We know that it's seasonally important habitat for all sorts of highly migratory species, tunas, billfish, and marine mammals, and so on. But we don't know a whole lot about the sort of the lower parts of the food web and what makes that favorable habitat for these higher predators. So there's a lot of interest in the shelf break right now, and I'm going to tell you a little bit about a survey that we did in 2010 to the shelf edge. So what we have here, I should have just gone straight to this figure to show it to you, but so this is the continental shelf edge, right? So the shelf itself is at depths, up to, call it, say, 100 meters or so. And then you have this very abrupt drop in depth to sort of 2,000 meters, and then a gradual decrease in the continental slope to the open ocean of sort of 4,000 meters. And this is a satellite imagery of sea surface temperature. So the color is showing temperature. And this is the Gulf Stream here. And then this is one of these eddies that's pinched off of the Gulf Stream and is bumping right up against the edge of the continental shelf. So what we did is we had a survey on the research vessel Connecticut where we did sort of studies at 4 locations. We looked at 2 canyons, Atlantis Canyon and Veach Canyon. So these are very steep canyons, much like a terrestrial canyon that bisect the continental shelf edge. And the hypothesis was that these canyons somehow serve to aggregate the krill and other plankton. So what we did is we surveyed these 2 canyons, and then for comparison we looked at places away from the canyons. And then because we had this warm core ring, we were able to also look at how the presence of the ring modulated that aggregative effect of the canyon. So we had the one canyon inside the ring, the one canyon outside, and then these other 2 sites also inside and outside of the ring. Okay, so just to show you some of what our operations look like at sea, this is deploying a few of the instruments. So this is one of our towed sleds that has our echo sounder, our fish finder on it. We tow it from a boom. It's deployed from, can't really see, but from a knuckle crane. So we tow that alongside the ship as the ship steams along. And then other instruments we deploy via the stern A-frame, like this cage here, which houses a video camera system. Most of these instruments are actually communicating up through the cable in real time to computers that we have in the main lab. So this is a photo of the main lab. These kinds of research vessels are sort of reconfigurable. When you go on board, you just have open benches, and we just install all of our instruments, and we install our grad students. And see here, this is the display of our echo sounders. So we're basically looking for the krill in real time, and then to verify that what we think on the sounder is indeed krill, we put our nets in the water. And so this is a little movie. So this net, unlike the bongo net, which is a fairly simplistic zooplankton net, effective for many species, just not so good for krill. What we have here is a multiple opening and closing net and environmental sensing system. So the environmental sensing, it can measure temperature and salinity and various other things. And the multiple opening and closing aspect, it actually has 9 nets on board. And we control it via a topside computer. So we can sequentially close one net, which then opens the next net. And what that lets us do is sample from different depth layers in the ocean so we can quantify, we can measure where the krill are in the water column.
So, to do all this and get boats and people, that must be expensive, which
Yes.
contributes to why you probably don't have a lot of data. Right?
No,
So-
yes, yes, exactly.
So given that,
That's very accurate.
given that, and given that there's a lot of navigational buoys, right?
Mhm.
Red nuns out there all over the place. Have, you explored or cost out, like setting up acoustic samplers on buoys, because they have all the other data being collected anyways.
Yes.
Just as a cheaper way to collect more data.
Yes, absolutely. So when I showed you the little picture of that torpedo-shaped vehicle, so there's all sorts of platforms, autonomous platforms like buoys, moorings, drifters, and we are trying to get these sort of devices on them. The problem is that they're really power hungry, so you need some kind of a power source like wind or solar, and even then it can be difficult to run these systems for long periods of time. But we're working on it actively because that is exactly the way to address the problem.
I could see that being an issue, but frankly, if the wind dies at night, you don't collect data for 8 hours, so what? Because you don't
Right.
have it today anyway, so.
Yeah. No, I mean, fair enough, exactly. So we've been focused mostly on these vehicles just because that was our sort of starting point, and the vehicles are nice, they're mobile. But getting things onto moorings is definitely a direction we're going in.
Can you tell us how heavy that thing is that you deployed with the nets?
Yeah, it's about 200 pounds.
Is that all?
Yep. Yeah, it's fairly light. It's yeah.
It looks much heavier than that. I suppose it's just me.
Yeah, it's certainly awkward, and they come in a variety of sizes. This is has a 1 meter squared mouth opening. They come in a little 1/4 meter, which you can definitely do from a dory, and they come in a 10 and a 20 meter, which is pretty big for a rigid. It's a rigid trawl.
That's true.
Yeah, so you need a big A-frame for that. And then again, we have these top predator observers who are rugged people that stay out all day long counting marine mammals and birds. So I mentioned this issue of the krill being able to avoid nets. What we've done to address that with our net system is we have an LED-based strobe light system. So this is our net here, and up top here, can't really see it, but there's a bank of LEDs, and they're flashing at very, very high light intensities. It's unpleasant for humans to look at it, and it's 1,000,000 times higher light levels than the krill are used to. And it's tuned to sort of a greenish wavelength, which is what the krill see best at. And it's aimed just in front of the net, and the idea is that it blinds them, stuns them, so that we can catch them. So we did some.
The acoustic thing is good for them, but the lights are like.
Yeah, exactly. So we did some tests through this layer that you see here in night and layer in the day, and we randomized whether we had the strobe light on or off. And we caught more than 10 times more krill when we had the strobe on during the day, and 5 times more when we had it on during the night. And you might say, well, what if you're just attracting the krill to the light? Because a lot of animals are attracted to lights. But what we did to address that is we made estimates of how many krill we thought were there based on the sounder. Compared that to the estimates we made with the nets when we had the strobe on, and they agreed very nicely, suggesting that we're not attracting them, we're just catching them at the abundances at which they're actually present. Okay, so in terms of the kinds of data we get from the sounder, just looking first at that Atlantis Canyon under the influence of the ring. So here's depth again and distance running off of the shelf edge. That's the continental shelf break, the bottom dropping off there. So we see layers of what look like zooplankton as we go past about the 200-meter isobath. And then here we're getting into the ring itself, and we start seeing a lot of stuff that looks like fish, though probably not commercial fish, more like lanternfish and weird mesopelagic fish. And then if we look at the cold canyon, the canyon outside of the ring, things look very different, a whole lot less of this fish kind of returns. And we start to see these very large aggregations of what looks a lot like plankton acoustically. These are on the order of 4 or 5 kilometers long in some places. And then when we sample these with our nets, we find that these are krill, so these enormous layers of krill, mostly in that cold canyon. So we can sort of sum up what we're seeing in the water column and map it out. So here the color of the dots, now the darker the color, the more krill. And basically at the two canyons is where we see the most krill, especially at the heads of the canyons, with about 10 times more krill at the head of that canyon outside of the warm core ring than the one influenced by the ring. So perhaps the ring is sort of washing some of the krill away. In both of the canyons, much more krill than at the places outside of the canyon. And I'm lumping together all of the krill, the different species. And I have a master's student who's looking at the actual composition of the different kinds of krill. You don't need to worry about the names, but basically because this is a boundary zone between the cold and the warm offshore waters, the species sort of can be divided into the ones that like cold water, and they tend to be really big, and those that like warm water. So the student has been looking at net tows that we did at the heads of the canyons and at the mouth of the canyons and at shallow and deep places away from the canyons. And you don't need to worry so much about what this is showing, just to say that the warm species tend to come up more in the presence of the warm core ring. That's not too surprising. But then he's also seeing big changes over very short distances. So comparing the catch at the head of the canyon to the mouth of the canyon, that's maybe 20 kilometers apart, there's a fairly large difference in the kinds of krill that are present. And what exactly is driving that remains an open question. And I should also add that the depth sounder on this ship wasn't working during this cruise. So we were trawling on the basis of the charts, which we're not very good at doing. And on 6 of the 12 hauls that we did, we hit the bottom with our little plankton net. And in many instances brought up all sorts of bottom-dwelling things, like in this case, 2 crab and then a piece of whale baleen. That's blue whale baleen. So we caught whale with a plankton net. Okay, so we have this team of observers counting the higher predators, and we can map out the distribution of where they saw various things. So this map is showing the distribution of toothed whales, mostly dolphins and sperm whales, which they only saw at the heads of these 2 canyons corresponding to where we saw the krill. I don't think they're feeding on the krill, but the suggestion is that there's something going on with the ecosystem there. And because this canyon was so interesting, we did a survey, a sort of what we called a shoelace survey, back and forth, up and down the canyon. And during that time period, the observers saw just all kinds of animals, so dolphins and baleen whales, rays, breaching, seabirds of various kinds, like these little petrels and these big sort of boils. So clearly something special going on in the canyon, and that's not really news. You know, they're obviously very important sites for fishing. But what we want to do is understand the mechanisms, and that's kind of the next step. This is a pilot survey motivating a more detailed study of, well, why are the canyon heads so important? If people have thoughts on that, I'm very curious to hear. In any case, I'll just sum up with a few conclusions. So hopefully I've convinced you that krill are important in the Gulf of Maine, that they have been underappreciated because they've been undersampled. And hopefully I've also convinced you that we've addressed those sampling issues so that we're now accurately capturing them. And then in terms of the shelf break area, we're finding that the krill are mostly found in these canyons and that there's something special going on with these multi-species assemblages. All right, so where we're going next, I have a postdoc who's looking at these stomach data and working with collaborators at the Northeast Fisheries Science Center to look at that in more detail and understand how these predator-prey interactions change over the years, how important krill is in some years versus other years, and trying to see if the abundance of various fish stocks track in time changes in the abundance of krill in the region. Again, we want to migrate this into this sort of ecosystem modeling in the context of ecosystem-based management. And although, again, we don't have a krill fishery in the Gulf of Maine, there is this Gulf of St. Lawrence krill stock that's going to need to be assessed. So we're working with colleagues at the Department of Fisheries and Oceans to get things like our strobe light system onto their nets, and working with them on some of these acoustic techniques. Okay, and just let me say a couple of things about ocean acidification. I think I got a few minutes, right? So how many of you are familiar with ocean acidification? All right, that's fantastic. That's a good representation. So as a brief review, the issue is that we have all this excess CO2 in the atmosphere. It's causing global warming through the greenhouse effect, but some fraction of that carbon dioxide is dissolving into the oceans, where it disrupts a series of chemical balances, leading to an increase in acidity, a reduction in pH. So ocean acidification is very much a global problem, but increasingly there's recognition that there's sort of hot spots, areas of the ocean that seem especially susceptible to acidification. And one of them, which you may have heard about, is the US West Coast. So I like to say that the governor of the state of Washington convened a blue ribbon panel on ocean acidification because it's thought to be strongly affecting their fisheries, their shell fisheries, that is. So the story there is that you naturally have this process of upwelling that's bringing waters that are low in pH just naturally. It's bringing them right up onto the shelf, right up into coastal areas where they're affecting the shellfish, because this excess carbon dioxide in the atmosphere dissolving in is just aggravating that natural upwelling of low pH waters. So in the Gulf of Maine, I mean, would you say that ocean acidification is a problem in the Gulf of Maine? I sort of didn't think it was until I started working with a colleague at Hui, Alec Wong, who has some very suggestive new data indicating that acidification may be more of a problem in the Gulf of Maine than we previously thought. So he published a paper just recently and had a press release and so on as well, based on a survey they did from New Hampshire down to Florida and into the Gulf of Mexico. And they actually found the lowest pH waters in that entire transit in the deep basins of the Gulf of Maine, particularly in Wilkinson Basin. And they have various ways of assessing the sensitivity to continued acidification, and the Gulf of Maine was the most sensitive to acidification. The consequences of this reduction in pH is, I mean, many animals in the ocean are sensitive to pH. But it's particularly concerning to things that form calcium carbonate shells. So things like our pteropods, but also things like shellfish and corals for that matter. And there's actually a depth in the ocean at which the water column becomes corrosive to calcium carbonate in its different forms. And in the open ocean, for these little pteropods, in the open ocean Atlantic, it's sort of 2,000 meters. It's very deep, well deeper than the range of these pteropods. But in Wilkinson Basin, the waters are verging on being corrosive in the sort of deepest parts of the basin. And the suspicion, and we have a series of cruises coming up this summer to test this, is that at other times of year, Alec collected these data in July, that at other times of year associated with this sort of seasonal cycle of productivity, that the water may actually be corrosive to certain forms of calcium carbonate, particularly what these little pteropods are made out of.
So you're saying more acidic the water sinks, basically?
So it's all about carbon dioxide, right? So carbon dioxide dissolves into the ocean from the surface, but what also happens is you have all the productivity, all the phytoplankton here, and that sinks. And then when it's down deep, and the flushing rates in the deep basins of the Gulf of Maine are sort of quite slow, you know, on the order of about a year. So all that biomass settling out hangs out here for a long while, where it's respired. The bacteria break it down, so they eat it and they produce carbon dioxide, and then that's what decreases the pH.
So areas near the shelf, then by nature, since the water will sink, will tend to be less acidic normally than shallower water like sections of the Caribbean.
In some cases, yeah, it varies with the exact currents and so on. In our areas, it's a sort of multifaceted problem because you have this natural process of this high productivity, and the Gulf of Maine is, of course, a very productive region, sinking being respired, producing carbon dioxide. You have ocean acidification, the enhanced CO2 in the atmosphere dissolving in. You also have coastal runoff, all of this sort of eutrophication, the biomass coming in from rivers that's also going to be sinking here, contributing to the pH. So some of it is sort of human influence, some of it is natural processes, but it all seems to be conspiring to reduce the pH. So the question is, how is it going to affect the animals? And we're thinking about that both in the context of our little pteropods, but also in the context of other animals that are sensitive to pH and important in the Gulf of Maine, like these krill. So I'll end there, and happy to take any questions, further questions. Thank you.
We'll try to get a light on so we can see your face for a minute.
Several slides back, you had a bullet point about the research you're doing, hopefully informing some policy moving forward. Can you speak to that? Do you have any idea, or are you not really there yet?
I'm not there, and that's, I don't know that I ever will be. Informing policy, that is. I sort of view it as I'll provide data that will inform the models, and then the people that do the models will talk with the managers and inform policy. So keeping it for myself at one step away, because I try and avoid conflict and politicization.
I think last year, the year before, a speaker, if I remember correctly, was making the observation like global warming is more the icebergs melting. There's more cold water coming down from the north.
Mm-hmm.
And that was sort of shutting off the Gulf Stream, entering the Gulf of Maine,
Mm-hmm.
and reducing the amount of nutrients entering, and therefore reducing the biomass.
Right.
Did I remember that right? And are you seeing that?
So you are remembering it right. There's sort of a natural cycle in whether waters come from the north or from the south, and then on top of that, there's the sort of runoff from the Arctic associated with climate change. That's contributing to this pH signal, because the Arctic waters, the cold waters, are also lower in pH. How it influences the krill, not quite sure yet.
You really highlighted how little is known and how much more. It's so clear from your enthusiasm how much more you'd like to know.
Right.
Can you make a comment on, you focus on a lot on the Gulf of Maine and also Gulf of St. Lawrence, but can you just sort of expound or just your thoughts about the rest of the, say, Atlantic U.S. shelf, Mid-Atlantic, how important are krill? What's your suspicion about krill in other locations that are important to, say, U.S. Atlantic fisheries?
Yeah, I think, I mean, on the West Coast and in Alaskan waters, they're very important. They're a key prey for pollock and so on in Alaskan waters. On the eastern seaboard, once you get south of sort of Hatteras, you don't see very many of the big species of krill. That warm, small, cold, big picture that I showed, those small ones, I think that they're important to the ecosystem, but not in the same way that these big, juicy ones are in the local waters. So I think we're at sort of the southern boundary of where it's a krill story. That's a guess.
Can I follow up? And the canyons and your studies there, do you think if it turns out to be a persistent aggregation of krill, would they be good places to have, say, long-term observations of krill dynamics?
Yes, absolutely. I actually cut out a slide because I didn't think I'd have time. But this ties into both your question and your question about long-term observations. So there's the National Science Foundation has funded what's called the Ocean Observatories Initiative, this enormously expensive observation program where they have a series of arrays throughout the world's oceans, and then one on the West Coast and one here just south of Woods Hole. So Woods Hole got $105 million to install this sensing array. Yes, it's a huge investment, obviously, and it's mostly focused on currents and the physical oceanography of the situation. So there will be some sounders on these moorings, and they have wind and solar and fuel cells, hydrogen fuel cells and so on to keep them going. It's going to be in place for 5 years, and then it's going to move to a different state. There was a bit of arm wrestling to see what state it would happen in, so it's off of our coast, but then it's going to move somewhere else along the eastern seaboard. In any case, this is located right here, which is not, it's not at a canyon, of course, but the box that you see here shows the range of these mobile, so-called mobile assets, the autonomous underwater vehicles, and they're actually going to be docking stations. Okay, so these things, this technology has already been proven. It works. Well, so far it works. So these things dock and charge up their batteries, download the data. The data gets piped up to a satellite, so you get it on your computer at home. They charge up. They fly for about a day, and then they charge up for about 6 days. So it takes quite a while to recharge. But these autonomous underwater vehicles are going to operate in this area, which goes just over to Alvin Canyon. Alvin Canyon is not named on the NOAA charts, but on our WHOI charts is named. And then what you see in this big box here is where the gliders. So gliders are more persistent vehicles. They actually soar like a glider. So they use a lot less power and have longer range, but they move much more slowly. So that box shows where the gliders are going to be operating, and that includes a couple of canyons, including Atlantis Canyon, which is why we went there on our survey, because we were trying to collect some sort of preliminary data. So they're wiring up this little box of the ocean with all these things that's going to include the canyons, mostly focused on physical oceanography, but we're certainly trying to get some biology into the equation.
Is there any plan, sorry,
Huh?
on that, which is pretty exciting. Is there any plan, you focused this, okay, it's oceanographic, but will there be any, let's say, the top predators, besides the in-water, you've got non-seabirds and turtles, could there be also different kinds of sensing above water and surface?
Yeah.
Plan on any of that?
Well, I mean, they're doing sort of air-sea exchange in meteorology and so on. And there's no plans for just observing. Yeah, actually, that's a good point. So one of the guys at WHOI has high-frequency radar that does, I think, look in a swath that includes this area. But it's sort of wide open. You know, NSF has really invested a huge sum of money in this, so it needs to produce, and so they're going to learn something about, you know, the dynamics of Gulf Stream meanders. But obviously, learning something about the fish stocks, things of sort of societal relevance, is something that NSF also wants to see happen. So the more we can tie it into fish and top predators, the better.
Kind of took the words out of my mouth. I was going to ask if aquatic drones are becoming more prevalent. Sounds like it might be a grand scheme.
Yeah.
Underway.
Yeah, it's
Above the air, under the water.
Yeah. I don't know if I might have cut that slide. I have a picture that we use. I cut it out when we try to get. No, here it is. So that's our vision of how things are going to go, and it's focused on these sounders because we're always trying to get more money to do our acoustic research. But so we have our underwater vehicles, we have our ships, we got our buoys. And then drones and piloted aircraft as well, but also drones.
Call them drones.
Oh, sorry, you're right.
Get permission.
It's really hard. Yeah, yeah. UAV. Pardon me. So we actually did look into this, you know, putting cameras, because you can get little model airplanes and you can give them smarts pretty cheaply. There's a guy at WHOI that, for $1,000, he builds these little things, which is, you know, almost expendable on the scale of WHOI sort of budgets. So anyhow, it has smarts, so it can you give it a GPS and it'll go out and fly a pattern. It can go way faster than the research vessels can. But the problem is getting the FAA to approve their use, because the small aircraft community doesn't like them. Understandably, doesn't like them. So his solution is he's working in the Antarctic, where there are no rules on this. If you work, beyond the 200-mile limit, you can also use them. Yeah, I think that these little airplanes are the way to go.
Any questions? Well, I think we'd all like to thank Gareth for coming up tonight. And I'll remind everybody, a lot of people wanted to come, and we have taped, as you see, we have professionals and semi-professionals here recording. So as soon as we can, we will post all 4 public seminars on the website. So probably keep an eye out on our www.tunelab.org. We're not sure yet, will it be Facebook side?
I don't know, but we'll have announcements on
We'll have
both
announcements.
sites
So
later.
if you missed any, they're extraordinary series, and particularly tonight, we really enjoyed it. And so tell people that couldn't come that these will be posted. And again, we really appreciate you coming. We've had incredibly informed audiences.
Yeah.
I think a high quality of questions, and we're really pleased with the science that we could help deliver to the community. So thank you, Dr. Lawson, and
My
thanks
pleasure.
everybody for a successful seminar series. And we'd also particularly like to thank Maritime Gloucester, Mary Kay's in the back, for hosting us, and we thank Borton's for also their support of our seminar series, and we hope we can continue. So again, thanks very much to our hosts. And thank you. One last thing, I'd like to thank Emily Chandler, our LPRC program manager, who is vital in putting this together. And I'd like to thank Charlie Blaney over there, and Henry Farini for loaning us the camera equipment. And thank you, Cape Ann community, for also taping. So again, thanks to all.